So welcome to the today's neuroscience seminar. I'm pleased to introduce Dr. Dale Lee, who is doing a lot of work on reward and what is the new area of neuroeconomics. He and I agreed we would avoid that term as much as possible, but nonetheless. Dale got an undergraduate degree in economics, and I left my notes, unfortunately, on my desk, but about 89, 88 in Seoul, and then came to the U.S., where he did a master's and PhD at the University of Illinois in Champaign with Joe Malpelli. So he did really down on the bare metal early visual system work, looking at LGN responses, and some very nice papers that I actually remember from those days. After that, he moved on and went from the sensory side to the motor side in Apostolos Georgopoulos' lab in about 95?
Yep.
Yeah, I was going to guess 94, but 95. Spent 2 years there working on movement, the beginnings of his interest, I think, in motor sequences was there, and had a very nice paper that, from my point of view, on neural coding issues about joint responses in neurons. I had done some work that people questioned, and when he and Apostolos got basically the same answers we did, it made it more acceptable somehow. After that, he moved on to Wake Forest, where he was an assistant professor for 2 years, and then in 2000 moved to the University of Rochester, where he really took off. He started doing a lot of work in frontal lobe. I don't know if that actually started at Wake Forest. It did.
He started in Rochester.
Started in Rochester and went back to his economic roots. Mm-hmm. I think in the way several of us have, but through the neurobiology of worrying how animals build values and make decisions. And his work since that time has really been on decision making and the neural substrates. In 1996, he was invited and accepted to take a tenured position as an associate professor at Yale, where he's in the Department of Neurobiology now. And they haven't put his title up, so I can't tell you. It's decision-making in primate cortical striatal networks. So welcome, Dale.
Okay. Well, thanks, Barry, for the nice introduction, and thank you for coming. So can I get my slide up? There we go. So that was a title slide. And as Barry mentioned, our lab's mostly interested in decision making, although there are some other projects that are going on right now. So that's what I'm going to tell you about mostly today. And before I get started, let me just acknowledge the people who did the work. The names at the top here, these are the people that have worked at my lab as either postdocs or graduate student. And the names in green colors are the ones that have contributed to this work. So I'm going to tell you largely about two sort of different types of experiments that we've been doing in our lab. The first one's about temporal discounting and intertemporal choice. And for that project, Xingyin Kai, Soyeon Kim, and Jaewon Wang had contributed a lot. And then in the second half, I'm going to tell you about some experiments that we've done on the line of reinforced learning and token economy. And Hyo Jung-seo was the primary driver for that experiment. So I'd like to start by giving you a really simple example that you have probably seen elsewhere many times too. So imagine that I'm giving you a choice between getting $100 with 100% certainty, right in front of you, versus 50% of making $300. Okay, and think about that. This is your real choice that you're going to make, and I'm going to give you the real money. What would you do? And think about what kind of criteria that you would use to make the choice in this case. Okay, so in fact, let's see. How many of you will take $100 hypothetically? How many of you will take 50 chance of making $300? So as you can see, it's not the kind of decision where there is a sort of unanimous agreement. People could have different sort of preferences, even for something simple like this. And that's what fascinates me. In other words, what's going on in our brain when you're trying to make a decision like this? And this has been obviously considered by many philosophers and economists and mathematicians and statisticians, people of all kinds of occupations. And I think if you sort of look at the book of history, the first name that comes to this issue is probably Pascal, who had proposed that you got to calculate the expected value. In other words, multiply, very simple manipulation. You just multiply the magnitude with the probability and choose whatever has the maximum expected value. You can call that an expected value principle. But then, soon after that, Bernoulli realized that that's not really what's going on in people's head, because you could have different risk preference. For example, in this case, imagine that what you really want to do after you're getting this money is to buy some product that you like. Like, for example, you like to buy a new iPhone or something like that. And the price of the iPhone is like $250. So $100 is not going to get you that. So then getting $100, even though it's 100% certain, is useless in that case, right? So when you have that kind of sort of nonlinear utility function, which can be incorporated into this term here, it may not be simply expected value. You may want to calculate what the economists call expected utility, and then choose whatever the option that gives you the expected utility. So that's pretty obvious. And then another sort of quantity that may come up in the studies of decision making is the concept of discounted utility. And this comes up when certain rewards are temporarily delayed. In other words, both of these hypothetical examples that we've considered so far assumes that these rewards are all immediate. But if you assume that some rewards are not immediately available and there's a time involved, after you choose something, you have to wait for a certain period of time to get the outcome, then the attraction or the value of that choice might be discounted according to this discount function, which is a function of time, the delay of the reward. So the notion is that if you the reward that you're expecting to get is delayed a lot along in the time dimension, then the value might diminish as a function of time. So you may calculate what they refer to as a discounted utility, and then choose the one that has a maximum quantity. And then another sort of concept that comes up a lot in the studies of decision making, especially in the line of reinforcement learning area, is a concept of value function. And what distinguishes value functions from all these other concepts that have predominantly sort of appeared in economics literature is that this is an empirical process, right? So if you, for example, change this question into getting $100 versus 300,000 won, that's the unit of Korean money, for example, and you don't know how valuable Korean monies are unless you are from Korea or something like that. And therefore, you may have to sort of make the choice and then empirically sort of estimate what you can do with that money, and then upgrade sort of your estimate of the value functions. Do you have a question?
Yeah, I was wondering if there's any variable other than time that's used for discounting in economic theory. I mean, the discounted utility function is based on time. Mm-hmm. Is there any other types of discounted utility functions?
Well, you can consider probability, effective probability as a probability discounting too. Or another possibility is that if you want to incorporate the concept of effort, the amount of work that you have to get the certain reward, then you can consider what people call effort discounting. So anything that can diminish the value of a reward through some, that's mediated by some factors, uncertainty, delay, or effort, anything like that you can consider as a discounting process. Does that help? Exactly. Okay, good. So this one is different because it sort of describes this time-evolving dynamic process where the value of a unfamiliar sort of state or object can be updated. So I think, I don't have to really get into the details, but this quantity that describes the difference between the reward that you receive and what you expected is known as a reward prediction error. And you're using that error to upgrade the value functions. So I sort of went through this briefly because I'm going to use some of these terms like discounted utilities and value functions and things like that throughout my talk to describe some features of the neural data that we get while we're doing an experiment to sort of understand how they're used and how they're computed in the brain that makes it possible for decision making. And another thing that I like to point out is that, regardless of which quantity that you choose, whether you're working with expected value, expected utility, or whatever, the reason why these quantities sort of feature heavily in the studies of decision making is because once you have these quantities are constructed or estimated, calculated, so that they have this property. In other words, you're going to choose the choice that has the maximum values of the quantities that we're talking about. So if you sort of, working with expected value, you're going to choose the one that has a maximum expected value. And when you have two choices, you can just make a comparison, or you can look at the difference between those two, and then choose the quantity that makes this difference positive. So I'm going to often refer to this as a difference in the value functions, or simply DV in some cases. But it's a really nice quantity to work with. In economics, people or decision makers will be sometimes considered rational or optimal if they can if their behaviors can be described by this simple rule. In other words, if you can construct utility functions that can express choices according to the simple rule. Okay? So as I mentioned in the beginning, I'm going to give you a couple of examples of studies that have been done using this kind of framework. And the first one that I'm going to talk about is intertemporal choice. And to give you a few examples, you can consider choice between getting $1, as it's very similar to the example that I mentioned in the beginning, versus getting $20 6 months later. And just like the risky choice example that I gave you, there is no correct answer. I mean, if you poll people, again, depending upon their time preference or patience, some people may just prefer to take $1 right now so that they can spend it right away, or some people may say $20 because I'm willing to wait. I don't need that money right away, and so on. So this is a subjective sort of process, and we're interested in as to how these types of information about the magnitude and timing information is processed in the brain and sort of leads to decision. You can do that with different kinds of food, or if you're a monkey participating in this experiment in a laboratory, you can do that with different amounts of apple juice. And just as another piece of background, this has been studied heavily in both economics and psychology. And when people started sort of working on this problem in 1930s, a guy named Paul Samuelson, who's a Nobel laureate in economics, basically proposed that the discount function that I mentioned earlier, in other words, the function that describes how steeply your subjective value of a reward goes down as a function of delay, might be sort of described by this exponential discounting function. But then several decades later, when psychologists actually started sort of examining this process in peoples and other animals such as pigeons and rats, what they found is that this is not the discount function that is observed empirically. What people and animals tend to do is that they tend to discount the value of the delayed reward according to the hyperbolic discount function, which is different from exponential discount function in that in the exponential, the discount rate, in other words, that's the fraction of the value that is lost per unit of time, is constant in the exponential discount function. That's the property of the exponential function itself. Whereas hyperbolic function has a steeper discounting for relatively short delay, and then it tends to flatten out compared to the exponential discount function. Okay? And the implication of that is that if you're a hyperbolic discounter, that means that you get more impulsive, and you sort of, your preference for delayed reward goes up when that reward is more immediately available, when that delay is relatively short, because that's when the delayed reward sort of escapes from the most heavy sort of discounting that happens, when the delay is relatively short. So, for a given sort of, when the overall rate of discounting is roughly the same, if you're a hyperbolic discounter, that tends to make you a little more impulsive. So this is nice sort of, contrast between the difference between these different forms of discount functions is nice because we can see whether, after you test your monkeys in a laboratory with an intertemporal choice task, you can sort of confirm as to whether the animals you're testing show exponential discounting or hyperbolic discounting, because lots of other studies in the past have predominantly shown that majority of them show hyperbolic discounting. So I'm going to show you some evidence that monkeys follow hyperbolic discounting later too. And I think many of you probably, since there's a lot of interesting, exciting work going on in awake-being monkey physiology at NIH, most of you are probably familiar with the kind of experiment that we do. But we almost exclusively work with rhesus monkeys and use, in all the experiments that I'm going to talk about today, we use eye movements as a behavioral measure. So we train the animals to indicate their choices by shifting their gaze either to the right or left, and then do some surgery so that we can put the electrodes into into different parts of the brain and then record the electrical discharges of individual neurons. So we do extracellular single-cell recording to try to correlate the kind of signals that we can hypothesize through these various mathematical models of decision-making, and then see how and where different neurons in the brain are encoding different components. So here's a intertemporal choice task that we use in our experiment. And like in many other experiments that I'm going to tell you today, at the beginning, you present a small fixation target, and it's at the center of the computer screen. So the animal begins the trial by fixating that target. And then in all the trials in this task, you always present one green target and one red target, and their positions randomized. So red target could be on the right side or left side in randomly selected trials. And the animal basically learns this relatively quickly in the beginning, that if they make an eye movement to the red target, they get 3 drops of apple juice, about 0.4 milliliter. And if they make an eye movement to the left, they get 2/3 of that. So they get 2 drops of apple juice, which corresponds to roughly 0.27 milliliter. And they learn pretty quickly that red is always better than green. So if you just present the red target and the green target, they start exclusively making eye movements to the red target. Then what we do is that we introduce these clocks. So these yellow dots around each of these targets is a clock, and indicates to the animal the amount of delay that will be imposed between the fixation of the peripheral target and the reward delivery. And in this example, if the animal, for example, chooses the red target, then there's going to be a 4-second delay since there are 4 yellow dots. If the animal chooses a green target, then there will be a 2-second delay. So in human terms, in this trial, the information that the animal is getting from the computer screen is that you could choose a red target on the left, get 3 drops of apple juice after 4-second delay, or you can choose the green target on the right and get 2 drops of apple juice after 2-second delay. That's the choice that the animal is getting. And they have to wait for 1 second until the central fixation light is extinguished, and they can choose whichever of these 2 targets they prefer. And then once the animal makes an eye movement, these yellow dots are extinguished one at a time, and that makes it pretty intuitive for the animal that these dots really are clocks. And then when all the yellow dots are extinguished, they get the juice. Does that make sense to everybody? Okay. So then the first thing that I like to show and have to show to you to convince that this is really intertemporal choice task in animals' behaviors, sort of what you would expect at least qualitatively before we can do any quantitative analysis on the behavior or do neural sort of analysis, is to show that this animal's choice in this task is what you'd expect from any sort of decision maker doing intertemporal choice task as you would expect. So what I'm plotting over here is the data from one monkey collected over 5 days. And the vertical axis is the probability that the animal is going to choose the small reward target. That's the green target. So if it's 0, that means the animal is always choosing the red target. If it's 1, that means the animal is always choosing the green target. As a function of delays for the 2 targets. So horizontal axis is the delay for the large reward target. And then these different colors are the delays for the small reward target. And for now, just ignore the lines. Just focus on the dots. Those are the real actual data that we got from this animal. And what you can see is that this probability is behaving pretty systematically. It goes up as the delay for the large reward goes up. So if you focus on the red one, for example, it goes up as TL delay increases. And then it goes down as the delay for the small reward increases, which is what you'd expect. In other words, animals preferring the small reward target more and more as the delay for the large reward goes up and therefore decreases attractiveness. And then it prefers the small reward target less and less when its delay increases and therefore reduces the attractiveness to the animal, right? And then so that's good because that tells you that animals sort of processing the information as you would expect, and then sort of those delay information is affecting the animal's preference in a systematic fashion. But we can show more than that because now what I'm going to tell you what these lines mean. These lines are the predictions from the best-fitting hyperbolic discount function for this data set. Combined with this softmax or logistics transformation, which I think is familiar with you if you have worked with decision-making problem at all, which just adds some noise to some randomness to the animal's data so that we can model this sort of gradual change in the animal's preference. And you can see that between the predictions of exponential model versus hyperbolic discount function, hyperbolic does a lot better. It's in fact substantially better. And the main difference comes from the fact that exponential model miserably underestimates the animal's preference for the small reward target when the small reward is immediately available without any delay. And that's exactly what you would predict for the difference between the hyperbolic discount function and the exponential discount function, as I mentioned earlier. So even though this is a relatively new task, and it's sort of done in a way that was not done in other experiments in the past, especially in the animals. It shows your sort of hallmark of sort of discount function that other people have observed in humans and animals. Okay. And then what we did was that we did this as a kind of sort of preliminary study to sort of make sure that the paradigm we're working with is working and it's producing some expected sort of features. But the one problem with this is that there's too many conditions. So anybody who's done awake-being monkey physiology will think that this is just way too complicated. So what we did was that we dramatically reduced the complexity of the task by subsampling the combinations of delays that we have. There are 45 different possible delay combinations here, and we reduced it to 7, I think. So but on the other hand, it's qualitatively the same task. Now I'm showing you the data from 2 animals that are collected during the neurophysiological recording. So here's same plot, plotted, the vertical axis being the probability the animal will choose the small reward target as a function of delay for the large reward and the delay for the small reward. And these are the data from the 2 monkeys that we tested originally. I'm also showing you the predictions from the exponential discount function in dotted line and the predictions from the hyperbolic discount function in solid line. And you can see that, as in the previous slide, even in this sort of somewhat limited set of conditions, you can see that the hyperbolic discount function is doing better. Okay? And then another sort of piece of behavioral data I'd like to show is that this is sort of the concern that we had before we started physiology, because it's nice. One advantage in using the clocks to indicate the delays to the animal during the intertemporal choice task is that you can vary randomly from trial to trial. That was the main motivation. Alternative way of doing it is to change the delay sort of slowly so that an animal learns the delay from the previous trials and that can use that information to estimate it. But then you're kind of stuck because you sort of have to choose the delay. You have to choose the delay that's similar to what was used before. But then the problem with this task is that the delay is confounded with the number of dots on a computer screen. Right? So, if the delay doubles, then there'll be twice as many dots on the screen, and that sort of, might be a problem if you're doing physiology. So one thing that we did was to dissociate the number of sort of dots on a computer screen and the delay. One thing that we did was to introduce 2 different colors. So, in a way, we're teaching an animal to do algebra with different bases, so that, just like before, a yellow dot corresponds to 1 second delay. But when there is a sign dot on a computer screen, each sign dot adds 4 second delay. So you sort of, to estimate what the delay of the reward is going to be for a given display like this, you have to count the number of yellow dots, and then count the number of sign dots and multiply that by 4, and then you have to add those 2 quantities. So it seemed pretty complicated, and then, we didn't know whether anyone was going to do this or not. But it turns out that if you use the same trick that we used in this task, where sort of gradually remove these dots after animal makes a choice, they learn this pretty quickly, and within about a month, they start showing sort of their preferences become almost indistinguishable between these different displays as long as the delay is the same. So here's again similar plots as what I showed you before, but now I'm using different symbols to indicate different clocks that have different number of sign dots. And you can see that if you, for example, focus on sort of cluster of dots like this, you can see that their behavior is relatively similar, regardless of the number of dots on a computer screen. So what the animal is really doing is to change their preferences based upon the delay of the reward. Okay, so then we have been recording, we started recording in this task in the dorsolateral prefrontal cortex for many reasons. One is that it's relatively easily accessible compared to some other structures that Okihira is, for example, dealing with that are very difficult to get to, at least for us. He makes it look very easy, but I think it's quite hard. But also because we know from the literature that in the dorsolateral prefrontal cortex there are reward-related signals. So this is a study, beautiful study from Watanabe's lab in 1996 to show that during the working memory task, neurons in the DLPFC can change their activity depending on the type of the reward that animal expects to get at the end of the trial. So we knew that DLPFC is more than working memory. It has reward-related functions. So we were wondering as to what these neurons might be doing during the intertemporal choice task. And here is one neuron that we got that clearly shows that there is something other than signals related to the animal's choice, something that's related to the difference in the discounted value. So this is a raster plot that shows the activity during the 1 second delay period where the target's already up on a computer screen. Animal's basically deciding whether he wants to choose a target on the right or left. And then there are different trials that are sorted according to this quantity that I mentioned in the beginning. That's the difference in the temporal discounted values for the two targets. So when DVL is simply the reward magnitude that animal expects to get from the leftward target, discounted as a function of its delay. And we can estimate this discount function from the animal's behavior, as I showed you earlier. And you can calculate that for both targets and detect the difference. That's what I call difference in the discounted values, or simply DV. And as I mentioned in the first slide, if this quantity is positive, that tells you that animal's better off choosing the leftward target, and if it's negative, then they should choose the rightward target, because it's a decision variable that the animals should use according to this model that we'll build. So the trials are sorted according to this quantity, and then there are two different colors of rasters. There are blue rasters and there are black rasters, and those simply indicate the actual choice that animal made. And you can see that there are a lot more black trials at the bottom, which are the trials where the animal chooses the leftward target, and then there are a lot more blue at the top. Okay? So this tells you two things. One is that, in fact, if you sort the trials according to this difference in the discounted values, that predicts animal choice. And then the other thing that this shows is that there is a gradual change, even within the black trials and the blue trials, that probably reflect this difference in the discounted value. It's hard to see that because you kind of have to do a lot of transformation in your head. So if I show you, then what I can do is that I can calculate the spike density functions for trials that are indicated by these different colors, and then plot them as a function of time separately for the trials where the animal is choosing the rightward target and the trials where the animal is choosing the leftward target. Then you can sort of, a little more easily maybe, see that there is a systematic change in the activity. But it's a lot easier to see that, obviously, if you average them, if you calculate the average activity during this 1 second, and plot that as a function of the same quantity that I used to sort these raster plots. So here, what's plotted on the vertical axis is the firing rate, average firing rate during the 1 second delay. And then horizontal axis is the same quantity that I've been talking about. And then empty symbols are the trials where the animal chooses the rightward target. Filled symbols are the trials where the animal chooses the leftward target. So you can see that there is definitely some effective choice. This neuron fires more when the animal is choosing the leftward target. But even within the same choice, you can see that there is some gradual systematic change in the neural activity, which is related to this decision variable. So we can say that in the DLPFC, neurons not only reflect the position of the target that the animal is going to choose, but their activity tends to be affected by decision variable, presumably the decision variable that the animal is using to make its choice. And then we did another control. So there are two different kinds of controls that we did in this task. One is something that I mentioned earlier to control for the number of dots. And the other one was to see whether we can, in fact, kind of turn off the decision-making process and have the animal do some other task that doesn't require them to compute these quantity decision variables, temporal discounted values. So one way to do that was to change the color of the fixation target from white, which means that the animal is doing the intertemporal choice task, into either green or red. So when these are done in randomized blocks, so when the animal is doing this control task in a separate block, the fixation color will be either green or red, and that simply tells the animal which target the animal has to select. So dots are meaningless, and the reward sign is always constant. So the temporally discounted values of these targets should be constant. The animal simply has to choose the target that has the same color as the fixation target. And when you do that, here's another neuron that shows this nice DV-related activity. So this is the one that is affected by the animal choice, sort of like very similar to the one that I showed you in the previous slide. And its activity is changing as a function of this delta DV, or difference in the temporal discounted values, especially for the trials where the animal is choosing the rightward target. But then when you look at the activity of the same neuron during the control trials where animal simply just didn't have to compute that, you can see that activity is much sort of flatter. In other words, effective delta DV or temporal discounted value is much weaker. Okay? So that's another evidence that this is not a simple response that's determined by the sort of visual features of the display, but it's something that animal might be computing to make its choice during the intertemporal choice task. Okay, but then, so that's what we expected to see. But then we saw, this is obvious, it happens all the time, I guess. We saw something more that we didn't suspect. In fact, this is something that's been observed in other parts of the prefrontal cortex. Podolsky and Assad's work in the OFC found something similar to this. Brian Lau and Paul Glimcher's work in the basal ganglia shows something similar to this. And that is that there are a lot of neurons in the DLPFC that is not, rather than reflecting the difference in the values of alternative choices, but the value of something that animal chose, okay? And in the literature, people are referring to this as a chosen value. That's the term that's been used in the previous studies that I mentioned. And we see the same signals in the DLPFC as well. So what I'm plotting over here is this is the same plot that I showed you in the previous slide, the firing rate as a function of delta dV. And you can see that now the slope of this function is different depending upon the animal's choice. So if the animal's choosing the left target, then it's a positive slope. If the animal's choosing the rightward target, then it's a negative slope. So what does that mean? It means that, depending upon the value of it raises the possibility that the value of the choice the animal makes might be influencing this activity, right? Because when the animal chooses the leftward target, activity goes up as a function of DVL minus DVR. So it has a positive correlation with DVL. But then when the animal chooses the rightward target, it has negative correlation with this quantity, which means that it might have a positive correlation with the DVR, which is the target that animal chooses. So to make it simple, we basically plot the same activity as a function of the discounted value of the target that's chosen by the animal. And you can see that the data basically line up much better when you plotted it as a function of DV chosen, suggesting that some neurons in the DLPFC encodes this sort of different signal between the values of the two targets that animal might be is choosing between, and other neurons might be coding the value of the chosen target, which could have definitely some different functional roles. Okay, so then more recently we've been recording from both caudate nucleus and ventral striatum of the monkey brain during the same task, because while we were actually doing this intertemporal choice, a couple of MRI papers came up, and they were indicating some interesting activities in the human striatum during the intertemporal choice task. And some of you might actually be familiar with this. There's some debate going on as to whether the brains are divided into hot system and cool system, and more rational part and more emotional part, and how they interact, for example, when you're making decisions and so on. So we were interested in sort of, seeing, examining the single neuron activity in different parts of the brain to sort of get more insights into those kinds of questions. And I think we don't really know the complete answers to them, but it looks like when you're recording from individual neurons in these different parts of the brain, the answers you get look a lot more complicated than when you're doing an fMRI experiment, which is not surprising. So these are the sort of rough division of the striatum into, caudate nucleus and the ventral striatum. And it's interesting to compare the activities between these two parts, because anatomy suggests that different parts of the striatum, especially the caudate nucleus and the ventral striatum, are connected to different parts of the brain. So caudate nucleus, for example, is much more strongly connected with the DLPFC, dorsolateral prefrontal cortex, whereas the ventral striatum is more closely connected to sort of more limbic cortical areas, such as anterior cingulate OFC or VMPFC. This is a slide from Susan Haber et al., which is a really nice anatomical study. So we basically repeated exactly the same experiments as I mentioned earlier, except that now we're recording from the caudate nucleus and the ventral striatum. And here's one neuron that we recorded from the caudate nucleus that has more or less the same feature that I mentioned earlier. In other words, when you sort the trials according to the difference in the temporally discounted values, you can see that black's at bottom, blue's at the top, and then even within the trials where the animal's choosing the leftward target, you can see that there is a gradual changes in neural activity, again suggesting that they might be coding the decision variable delta dV. And again, you can see that probably more easily if you look at the average firing rate during the delay period, which changes a function of delta dV. So neuron fires more when the value, temporally discounted value of the left target is greater than the rightward target. And again, this neuron loses that value-sensitive activity if the animal is doing the control task where they're simply choosing the target that has the same color as the fixation target. And just like in the DLPFC, we also see in the caudate nucleus signals that appear to be related to the temporal discounted value of the chosen target, right? So when you plot the activity as a function of delta dV, you get this nonlinear relationship, which gets lost again during the control trials. But then when you plot it as a function of dV chosen, or chosen value, you get much nicer relationship, again suggesting that similar to what, as I mentioned earlier, people in Glimch's lab have found, some neurons in the coded nucleus might be coding the value of the chosen target. And I sort of mentioned this all briefly in the beginning, and it's really fascinating to think about the relationship between delta dV signal and the chosen value, because they really like chicken and egg problem, which one comes first, right? You sort of have, the idea is that in order to make a choice, you sort of have to compute the difference between the dV, because you're going to choose the one that has a larger value. So you need that signal before you calculate. But then, in a way, so this is a signal that you might think can appear only after you make a choice. But as I'm going to show you later, and this is what's also shown in Padua-Schioppa's and Assad's study in the UFC, that in the brain, these signals actually appear relatively early. So I'm going a little bit ahead of myself, but think about sort of, keep in mind that the timing of these two different kinds of signals, delta dV and the dV chosen, might tell you something interesting about the brain. Okay, and here's a population summary. So these are the fraction of the neurons in these three different areas that I showed you, DL, PFC, and the coded and the ventral striatum, that are related to this different signal and then the values of the target that's chosen by the animal. So you can see that the coded roughly, there's a, there's some, one way to simplify it and then sort of understand it is that if you compare, and this is, I didn't mention, this is the fraction of neurons that are showing activity that's related to choice, whether the animal is choosing the rightward target or leftward target. All of these variables are estimated in the regression model that has all these three terms. And you can see that overall, the DLPFs and CD coded nucleus look more similar than the ventral striatum, because here, these two areas look similar, and the ventral striatum is a little bit lower, and here, the DLPF is a little bit higher, but you can see that the TV chosen signal is more stronger in the VS. But if you compare CD and the VS, it's one thing that sort of comes out of this is that the VS ventral striatum is really, really about the value of the chosen target. There are only very few neurons that are responding differently depending on whether the animal is choosing the rightward target or leftward target. And they don't really seem to care about the difference between the values of the two targets, but there are a lot of neurons in the VS that are related to the temporal discounted value of the chosen target. And that this research has not been really sort of shown clearly. MRI studies really haven't distinguished between these two types of signals. So it'll be kind of interesting to see whether we can make the same observations humans if you sort of design the task properly so that you can dissociate these in the fMRI signals, fMRI experiments also. And then as I mentioned earlier, we can look at these signals as a function of time, because you can take the same multiple linear regression model and then apply that using a sliding window to see how these signals change as a function of time. And basically, the pictures show the same thing. So if you look at the time course of the signals that are related to the animal's choice, or the delta dV signal, or the chosen value signal, these two areas, there's some differences, but show roughly all of these signals are present in the DLPFs and the coded nucleus, whereas if you look at the ventral striatum, it's predominantly the dV chosen. But as I mentioned earlier, look at the time course. This signal is not coming, at the end of the trial. Basically, it's coming up within about 20 milliseconds after target onset. So as soon as the target comes up, in a way, the animal has already decided. And the time course of this signal is very similar to the time course of this blue, which is the difference in the temporal discounted value. If you superimpose it here, for example, you can see that they sort of kind of go up at the same time, suggesting that even though, in theory, you can dissociate these different kinds of signals in the brain, they kind of sort of might evolve almost simultaneously. I think it'll take a lot more thinking on our part as to what the relationship between these two signals might be. And the fact that it sort of, there's an anatomical sort of segregation in that dV chosen is much more concentrated in the ventral striatum, I think is also interesting, something that we're thinking a lot about these days to figure out exactly what it means. Okay, so then Soyeon Kim, who's the postdoc who sort of really spearheaded this effort in this project, has recently been working on a modified, new, somewhat different intertemporal choice task, which has an uncertainty in it, because I mentioned earlier that probably the two sort of the themes that are most frequently considered in the literature is something about uncertainty and something about the delay. And in real life, obviously, in many decision-making situations, you kind of have to consider both of them. So it will be kind of interesting to understand where in the brain these two signals are combined into a sort of coherent quantity. And to do that, we sort of decided to look at a task where you can manipulate the delay and uncertainty independently. And it's, we call that intertemporal gamble. And so what we have in this experiment is that we have two different kinds of trials. And in the first type of trials, everything goes exactly what I described earlier. So this is half of the trials in this intertemporal gamble is pure intertemporal choice task where there is no uncertainty. Animal always gets the reward that is promised at the beginning of the trial, either 2 drops of juice or 3 drops of juice after variable delays. But then in the other half of the trials, what we have is that trials are very similar to this delay intertemporal choice without any uncertainty. The only difference between these two trial types is that the larger reward target is now signaled by the cyan color. Okay? So when the animal sees green target and a cyan color on a computer screen, that tells the animal that he has a choice again between 2 drops of apple juice and 3 drops of apple juice after variable delays that are indicated by these yellow dots. But when the animal chooses the larger reward target, i.e. the cyan dot, the reward is delivered only 50% of the trials. So by comparing the activities in all of these trials, we can look at the effective delay and how that affects the animal's choice. And by comparing the activity between these two cases, especially when the animals choose the same target, we can then assess the effect of uncertainty, how that sort of affects the animal's choice. So again, same thing. It's one thing to do these kinds of manipulations, and then you sort of have to show that really affects the animal's choice systematically. So what I'm plotting, I'm going to show you the same kind of graph that I showed you earlier. Now the vertical axis is the probability of choosing the small reward target just like before. Horizontal axis, the delay for the large reward just like before. But now the graphs that you're going to see in this plot will be a little more complicated because now we have twice as many conditions. But that's why I'm introducing it sort of one at a time. So this is the plot. Again, just focus on the symbols first because those are the actual data. And this is how the animal's choice of the small reward target is affected by delay for the large reward when that small reward is immediately available without any delay. So it goes up as a function of the delay of the large reward as you expect. And then if you delay the reward small reward that you're going to get, then obviously that reduces the attraction of that target. So the probability of choosing a small reward goes down just as you'd expect. And then now you can sort of guess as to what these graphs will look like if the large reward becomes uncertain. That should reduce the attractiveness of the large reward targets. The whole curve, though both of these curves should be shifted upward, and that's what happens. So dotted lines are the same thing as the solid line, except that the large reward now has a 50% probability. Okay? And the nice thing about this is that it's, obviously everything's sort of behaving predictably as you would expect qualitatively. But nice thing is that you can also fit a relatively nice econometric model to get a really sort of quantitative handle about how the animal's preference is changing. So I'm using the most simple model that you can possibly imagine, where I'm expressing the discounted utility for a reward that has a magnitude of x, delay d, and probability p as a function of product of these three quantities, magnitude. So I'm assuming a linear utility, if you, for those of you who sort of understands the difference between utility and the magnitude. And then this is the discount function that I mentioned earlier. And then this is the probability weighting function. So we're assuming that animal may not know that it's exactly 50%. What does the animal think? What the real reward probability is for the large reward target? We can estimate that from this model. And if you assume in hyperbolic discount function that becomes this, and since we were testing only 3 probabilities, we were testing only one probability, 0.5. So we can't estimate this whole function. What we can do is that we can estimate the probability weighting function when the probability is 0.5. So we can estimate this single parameter from this model. And the lines that I showed you already are the predictions from this simple model that has two free parameters. And you can see that, considering the number of conditions that we have, it's doing a pretty good job. And that's not surprising. What's really interesting is the fact that we can sort of read off from this data as to how patient the animal is. That corresponds to this parameter k, which is in the discount function, and also what the animal's risk preference is. In other words, what's the probability that animal thinks that he's going to get the reward when the animal chooses when he chooses the large reward when that probability is in fact 50%, this fitting that I think that's the best sort of the best part of this model fitting exercise, that you can find out that animal thinks that, according to this model, thinks that the reward is coming 77% of the time, suggesting that animals actually seeking, it's preferring, is preferring. If you define risk neutral as a 50%, right? Because that's when the animal really said the risk is coming, the reward is coming 50% of the time. So we've been, again, recording, we're still recording from the DLPFC in this task. And again, the first question they're asking is the same as before. In other words, can we find the signal, in fact, let's go back, that's proportional to this. In other words, can we find the signal that's really best predicting the animal's choice according to this model in DLPFC or any parts of the brain? And the answer is yes. But there's a little more than that. So let me first show you an example that's consistent with what we expected. And this is the plot that's sort of similar to what I showed you before, except that now all these trials are from the trials where the larger all of these are trials from trials where the larger reward is certain. So this is exactly the same intertemporal choice that you saw before. And you can, again, see the same thing, that there is a difference in the black and blue, and then there's some gradual change in the activity even within the same choice. And we can do this now since we have both risky larger reward target and a certain reward target. I can show you the data from the trials where the larger reward is uncertain. And you can see that there are a lot more blacks here than here, which is, I'm sorry, that's not the case. So in this case, I was choosing the small reward a lot more often. But since the left and right side of the target positions are all randomized, you can see roughly half and half in black and blue. But you can see that the way that activity changes as a function of discounted utility. In this case, the quantity that I use to sort these trials is the discounted utility. That includes both the probability and delay in it. And you can see that those two trials look roughly the same. Okay? And you can also confirm that by plotting the average firing rate during this 1 second as a function of the difference in the discounted utility. So just to remind you, discounted utility is this quantity here, the one that is proportional to the reward magnitude, but is discounted both by its uncertainty and delay. And that seems to be a good predictor of the activity of neurons for something like this. But then this wasn't the case in all the neurons that we got from the DLPFC. Again, so the rasters are plotted in the same way, and you can see that it's a bit more complicated. There seems to be much more difference between these two groups of trials, and that becomes really obvious when you plot the average firing rate as a function of this delta du. Because now this one's sort of like what you'd expect. So if you just did the sort of deterministic, non-probabilistic intertemporal choice that we did in the beginning, you'd think that this neuron's just calculating the values for the animals so that it can make a choice. But when the larger group becomes uncertain, that you can see that relationship is broken. So it's doing something more complicated. And right now, we're still recording from these from this task, so we haven't really had a lot of opportunity to sit down and think a lot about what these neurons might be doing, but we think that it's a sort of evidence of a sort of pre-decision process where the animals are still processing information about uncertainty and delay information somewhat in parallel before they get all merged into the kinds of neurons that I showed you in the previous slide. Okay, and this is again, population summary. So for the data that we have so far, a fraction of neurons that are related to simply the animal's choice or delta discounted utility. So this would be the decision signals that you would be using to make a choice in this best fitting model. And then these are the fraction of neurons where that delta du interacts with the animal's choice or uncertainty. So this is probably related to chosen value, as I mentioned earlier, because this shows you that the slope of the neural activity as a function of delta du is changing depending upon the animal's choice. But this is the one that we're still scratching our head about. And you can see that that's actually has the largest value in this plot. So we really have no idea, no clue so far as to how these signals, what the functional significance of the signal is in the DLPFC. I hope to figure that out soon. Okay, so those are the sort of examples of experiments that we're doing in terms of intertemporal choice and in the framework of discounted utility models. And then in the remaining time, I'd like to tell a little bit about one other sort of line of research that we're doing in the lab that is more along the spirit of reinforced learning. So as I mentioned in the beginning, these values are in some cases, you have all the information that you need to calculate the value functions in the intertemporal choice. If I basically tell you that animal's choosing between small reward, large reward that has certain delays, and the large reward has certain probabilities, that's all you need to know to make a decision according to your favorite decision-making model. Whereas in real life, you don't know the value of the things that you're going to get. You just have to try it out and sort of figure out what the value of taking a certain action is. And this is, during that sort of learning process, many different things could happen, and according to this simple sort of scheme that I found from a sort of textbook on experimental psychology, people have classified the kind of learning that could occur in this context into these 4 different categories, depending upon whether the frequency of behavior increases or decreases after when the stimulus is presented versus omitted. So this is what we all know, positive reinforcement. When you increase your probability of doing something after you get a particular feedback. We all know this too. So this is when you decrease your probability of doing something after you get certain kind of feedback. These two are less familiar, I think. This is where you increase the behavior of doing something, you increase the probability of doing something when something disappears. That's what's called negative reinforcement. And I think this is probably the least familiar to you. Negative punishment, which is you decrease the behavior of doing something, you decrease the probability of doing something when you lose something. Okay? So I think society depends a lot on negative negative punishment, because they give parking tickets, which takes money away from your bank account when you park illegally. People and animals don't like losing something that they value, and that could definitely have a sort of inhibitory, that could suppress certain kinds of behavior. But surprisingly, this has been relatively almost never looked at systematically in the laboratories, even in behavioral studies. Which we found out after we finished the study. In economics, obviously these are commonly referred to as gain and losses. So we went ahead and sort of designed a task where you can look at the processes that are related to both positive reinforcement and negative punishment in the same experiment. And to do that, we designed the task that uses token economy. So instead of rewarding an animal, the idea here is that we wanted to do something where you can compare the processes, behavioral and neural processes that are related to positive reinforcement and negative punishment without really introducing aversive stimuli to the animal. And you could do that basically by using a token. So instead of getting reward or juicy reward at the end of the successful trial, in this decision-making task, if the animal makes a choice, and if you want to reinforce that choice, what we do is to present a token, which is this red disc. So there's 6 red circles on a computer screen, and they can filled up or they could get emptied, which corresponds to gain and loss trials. And in some cases, you could just basically have a neutral feedback and don't change the number of tokens on a computer screen. An animal does this repeatedly, and the reason why this is a gain and loss is because whenever the animal acquires 6 tokens, when the animal acquires the 6th token, they're automatically exchanged with the 6 drops of juice. So it's a kind of conditioned reinforcement, just like money. You and I could consider as money as. And there are some detail that I'm going to skip for the sake of time. This is a paradigm that our labs have been using for about 6 years now, and we published several papers on this. And it basically uses the competitive game as a framework. Just to briefly, this is like binary version of rock, paper, scissors, where there is no right or correct answer. And the reason why we do this is because you wanted to have some stochasticity in the animal's choice behavior. So if you create a payoff matrix like this, where the animal is going to get a token when its choice is matched by the computer's choice, and either doesn't get any token, a neutral outcome, or loses a token when the computer chooses one target and the animal chooses another, then this is a very basic sort of part of game theory. You can calculate what game theory is referred to as Nash equilibrium. If you saw the movie Beautiful Mind, this is the contribution of Nash, which tells you what the optimal choice probability should be. So for this particular payoff matrix, if the animal is behaving optimally, then they should choose the safe target that has no loss with 2/3 probability, and then choose this risky target that has a loss with 1/3 probability. And we can track the animal's behavior while changing the position of the risky target and safe target from block to block, just to show that animals actually doing this reasonably close to optimum. And we can also look at their outcome probabilities, and these are results from 3 different animals, and you can see that there are some systematic differences, which is something, again, I'm going to skip, but that's we can do a lot of interesting analysis to try to understand what kind of algorithms animals using to show this behavior. But the important thing here is that there's some randomness involved. Animals are going to choose rightward target or leftward target with some probabilities, and then their outcomes are unpredictable. And this shows you that when the block changes, this is the Nash equilibrium for the previous block, and then this is Nash equilibrium for the next block. And you can see that within a few trials, animal can switch to new optimal strategy almost completely. And then another interesting thing. So this is the sort of the most exciting part of the behavioral data from this task, which just shows you that after animal gains a token, after choosing a particular target, so say that animal chose the rightward target and that led to the gain of a token, then that increased the probability the animal is going to choose the same target in the subsequent trials, maybe up to 5 or 6 trials. That's what these blue symbols are showing. Those are the regression coefficients from a logistic regression model that estimates the effective gain, previous gain on the animal's choice in the future. So that's expected. In other words, since tokens are condition reinforcement, they have reinforcing properties. It's almost tautology. What's interesting is that when the animal loses a token, it's punishing. And therefore, if the animal chooses a particular target and then loses a token, then it decreases the probability the animal is going to choose the same target in the next trial and increase the probability the animal is going to choose the other target. And that's shown by these red symbols. And some of you might have already noticed that there's some idiosyncratic changes in the lag1 parameter, but besides that, they're all largely negative, again suggesting that losing a token is a punishing to the animal, which surprisingly to me has never been documented in the animal learning literature. So I'm saying this actually whenever I can, so that I can get corrected. But so far, I haven't found any example in the literature of this type. And then we recorded from several different areas in the brain. In fact, I'm showing you 3 different areas that have the results from which are just published, but we also recorded from caudate nucleus in the ventral striatum, and recently we were recording from amygdala. And I'm going to just show you the fact that since I'm running out of time, that in many of these areas, we find outcome-related activity that are different for gains and losses. So here's one neuron that we recorded from the dorsomedial frontal cortex, where during the feedback period, so this is when the animal receives a signal that it's going to gain a token or lose a token. If it's a neutral trials, that's the activity in green. And if the animal loses a token, then activity goes up. And if it gains a token, the activity goes down. And it's unaffected by the animal's choice. So these are sorted according to where the animal is choosing the leftward target or rightward target. And you can see that they're relatively independent of the target. I'll skip this part. And here is a neuron, another neuron in the DMPFC, that is actually a bit more complicated because here the outcome signals are integrated into the animal's choice. So whether the animal is going to increase, the neuron's increasing activity for gain trials or not depends upon the animal's choice. And this is important because this is the kind of signal that you need to update the value functions. In other words, just know that outcome was good or bad doesn't tell you how you should modify your choice. You have to know what you did that leads to that outcome. So these are the neurons that are signaling the conjunction of your own action and its outcome, and therefore it's critical to update the value functions properly. And these are the summary of 5 areas that I mentioned earlier, showing you the fraction of neurons that are changing their activity after gain feedback, loss feedback. And then these are the fraction of neurons that are changing their activity according to the conjunction of the action and outcome. More important, these sort of affects the mood. You feel good if there's a gain, but this is what you have to do in order to update the value functions properly. And you can see that the DMPFC sort of stands out, and then caudate nucleus actually has a lot of signals that are related to these conjunctions. And briefly, this is sort of, I think it's penultimate slide. We've been doing the same experiments in the amygdala. We have about 30 neurons now. And a lot of the neurons in the amygdala also, not surprisingly, Daniel Saltzman showed us, and before that, there were already several studies to show the outcome-related activity in the amygdala. We were looking for something more complicated, but so far, the main thing, probably the only thing that amygdala seems to care appears to be this outcome, and doesn't have other sort of more complicated signals. but there are a lot of them. So here's a same plot that I showed you earlier, but now we added the amygdala, and you can see that there was a lot of neurons that are changing their activity as a function of gain, a lot of neurons that are, more than any other brain areas that we looked at, a lot of neurons that are including changing their activity after loss, but they don't seem to have this more cognitive sort of signals in the amygdala, which is what we were looking for. So we were disappointed, but at this point, we don't know what to do, whether we should just stop or whether we should continue to just document this in the literature or not. So any feedback would be appreciated. And then there was one other study that we were doing in the lab to look at the effect of hypothetical outcomes, but I'll skip that since I ran out of time. So these are the conclusions. We're basically making some progress about how the value signals are represented in the brain, how that's computed from these various sources, including probability, uncertainty, and magnitude. And the experiment that I didn't have time to get into was looking at the activity related to hypothetical outcome, which we also find in the DLPFC. Okay, thanks for your attention.
We have time for a few questions.
Did you find any difference in the behavior based in gender?
No, because we tested only, so this gets to an interesting question, interesting problem that we had. So I intended to buy only the male monkeys just to reduce the uncontrolled factors. But it turns out that one of the monkeys that we used in the token experiment was a female. And the vendor didn't know it. The vets didn't know it. We didn't know it until we were almost done with the recording. So there was one female and two male monkeys in one of the studies that we did, but we didn't find anything different. There was some difference in that. I can actually go back to this slide because it's sort of interesting. This was the one where there was a behavioral data from 3 different monkeys. So monkey K was the female monkey. And you can see that this monkey, the animal is actually shows the most robust effect of token loss in LAG1. So this animal basically hated it. If she chooses a target and loses a token, it reacted more negatively than these other 2 male monkeys. That's the only comment that I can make. Yes.
So I still want to ask how the DB choice signal helps
Mm-hmm.
people to make the decision. You said that signal is really early, but I'm still not sure I understand how it can be useful.
Right. So here's how I didn't sort of go into that in details, but here's how you could think about it. So, in our case, the position of the large reward and small reward, or uncertain reward and certain reward, are sort of appearing in random positions. So this sort of relationship between these two different kinds of signals will become more interesting if you think about the sort of the frame of reference in which you think that decision might take place. In other words, do you compute the value of the target on the right and then value of the target on the left and then compare them and choose whatever has a greater value? Or that's one possibility. Another possibility is, do you calculate the value of large reward and the value of the small reward, and then look at the difference, and then decide whether you're going to choose a large reward, small reward, and then look for the screen again and see where the large reward is, and then choose that. Did you get that? In other words, the second way, it sounds more roundabout way of doing things, but it could be more general, because a lot of the choices that we make are not spatial. Right? So this is why, if you remember Podolsky and Assad's paper, they make a big deal about sort of action-independent sort of evaluation, because in their case, the animal was choosing 2 different kinds of juice, and although they're appearing in random positions, what might be going on in the OFC, for example, might be that animals calculating the value of the apple juice and the value of the orange juice, and then choose which type of juice the animal is going to choose, and then based upon their values, obviously, and then find out where the apple juice is, where the juice reward is to make an eye movement. And you could, since this is independent of space, and space is just one of many dimensions that you have to consider. If you think that decision-making process in the brain, sort of the core decision-making process, is somewhat independent of space, then the comparison of the values independent of sort of their spatial positions might be sort of more sort of primary, and therefore the value of the chosen signal might be actually what's driving us to what actions you're going to produce. Does that make sense to you? I mean, it's a speculation, and we don't really, I don't think any studies ever sort of resolve the difference between these two possibilities, but I think it remains as a really fascinating question that I think people should look at.
In most of your tasks, the animals choose between one or two outcomes that have different rewards associated with them.
Right.
Is there a fraction of trials upon which they look somewhere completely different and get no reward at all? Or are there any other contingencies, or do 100% of all trials of monkeys do fall into these?
Well, the only exception would be, well, so there are several, I don't know where you're getting at, but in the intertemporal choice task in the second version, there are uncertain trials where if the animal chooses a large reward target, the reward is delivered only 50% of the time. So there are cases where the animal doesn't get reward in that case.
No, but whether they can do anything about, there's two targets here and here.
Right.
Presumably they have fixation windows around them. And if the monkey just looks away, looks off somewhere completely different, what happens to that trial?
Oh, then they don't get the juice. It's aborted.
And what fraction of trials fall into that? In other words, if to estimate what is the optimal behavior,
Right.
one thing they have to factor in is what's the possibility that there's a third outcome that they have to factor in.
That's right.
And we
So,
have
you
to
know,
factor in too.
that's right. So, all of these cases, the error rate's relatively low. In the token experiment, I don't have a slide here, but this is sort of, in a way, what Barry's sort of, looking deeply into. If you look at the error rate as a function of the number of tokens that animal has, it goes down monotonically. It shows you, this is, in fact, in a paper that just came out. So if I remember correctly, if the animal has a no token, in some cases, if the animal is going to take a long break, they usually do that when they lose all the tokens that they have. And then the error rate, on average, when there is no tokens on a computer screen, is about 20%, and it goes down monotonically as the animal acquires more tokens. So number of tokens in our case definitely has a role in affecting the animal's motivation to the performance, if that's what you're getting at. And there are a lot of, I mean, this task allows you to look at many different kinds of signals, one of which is signals that are related to the animal's motivation, presumably how many tokens that it has. And in all the cortical areas that we looked at, I think the percentage of neurons that are modulating their activity according to the number of tokens is upward 40 and 50%. So it's a very robust signal that's present, as Barry has shown in many different areas. That's a signal that you encounter very frequently.
So,
So is the error rate in the first test so low that the difference between the value of the chosen object and the value of the large and small reward is essentially indistinguishable?
I'm sorry, say that again?
The value of the chosen target,
Right?
the value of the red and green target,
Yes.
that difference, are those essentially indistinguishable because the error rate is so low?
I don't understand your question.
Aren't those two different quantities? The value of the difference in value between
Oh, well, yes. Well, so in general, so you're asking me whether the value of the chosen reward can be separated by the size of the reward. Is that what you're getting at? So, if you look at the conditions that we have, the large reward tends to have a longer delay. So on average, I mean, we titrated. The reason why we chose 2 drops versus 3 drops is because we wanted the animal to choose the larger and smaller reward roughly half of the times. So on average, their discounted values, presumably the subjective value of those 2 different rewards to the animal are roughly the same, but they will not be exactly the same.
so thank you very much, Taylor.
Okay, thanks.